Hello everyone, welcome back. Um, as I mentioned with the lymphocytes, they can either attack things directly or they can make these things called antibodies. And I'm sure everybody heard of this term, antibody, antigens, and so forth. So, um, in my experience, a lot of students don't know the difference between antibody and antigen, and that's really important that we learn this. So during COVID or post-COVID age, it's important that we learn this, and you have to understand how antibodies and antigens work in terms of vaccines and in terms of being infected, in terms of clearing it. And the first thing to do here is let's talk about the definition of antigen. So antigen is anything that's not yourself. So it can be anything that's outside of your body. It could be a cancer cell, and your body doesn't recognize it. It could be pollen, it could be dust, it could be basically anything your body just doesn't recognize it. And this one here is going to then trigger the immune response, and the antigen is not part of your body, so it's going to trigger your body to make these things called antibodies. So maybe a cell, so antigen can be a cell from another individual, a cell from invading microorganism. So your body is going to make antibodies in response to the antigen, and antibody is a type of protein. So plasma has antibodies, or we call them globulins before. So I'm gonna say these globulins are a type of protein that recognize foreign antigens. So these proteins will go and find antigens, so recognize the antigens, and then they bind to the antigen and they find the right kind of antigen. So we'll go ahead and stick to them, and we can see that usually the antigen antibodies are Y-shaped, so this shape. So that's a common shape you might see, but they're not all Y-shaped. So when they go find an antigen that's something that doesn't belong in your body, it goes and grabs onto it and starts making these clots called, and then it causes agglutination. Agglutination means it's, just kind of clot up and clump together, and then your body takes care of it. So what you want to uh, have is antibodies to go find an antigen and then kind of take it down. That's the reason it's called antigen, and these are called antibodies. So going back, the term antigen here, gen means to generate. So the word gen rating, and then the term anti means antibody. So that's where the names come from. So this antigen is antibody generating molecule. Whenever your body sees antigens, it's going to start generating antibodies, and the word gen means generating, anti means antibodies. So when you put it all together, antigen is going to generate antibodies, meaning if you get dust or dirt in your blood, you're going to, or you get hit with a vaccine, that's going to introduce antigens in your body, and that's going to cause the body to start making antibodies and start cranking out these antibodies and take care of the foreign object. So the difference between antigen and antibodies, as I said before, all antibodies, they have various shapes. Most of the common shape is the Y shape. So if you study immunology, and then you'll learn that there are many different types of shapes for the antibodies. So most common type is Y shape, and that's going to be these guys here. But there are other shapes. So let's see. So those are some other shapes that you can find in the antibodies. And these are called, so you have IgW, IgM. The term Ig stands for immunoglobulin. And then this is part of your immune response. So globin is part of the protein. Immune means the immune system. So basically, they're named after the letters of alphabet. And you have IgM, IgT, IgD, various, different ones. And the term Ig is same in all of them, which means immunoglobulin. So if you ask an immunologist about these, they'll know exactly what you're talking about if you just were to name one of these antibodies. Okay. So antibodies bind to the antigens and destroy them. If antibodies bind to foreign RBC, then clumping or agglutination occurs. Agglutination means clumping. What I mean by that, we have all different types of blood types. So we are born with blood types either, type A, type B, type AB, or type O. So I'll go through these now. So if you're going to be type A, that's what this is representing. Is that your red blood cells? You got this protein that's going to be called the surface antigen. So remember we said antigen is something that generates antibodies. So this antigen doesn't do anything for yourself because you're born with it. So if you're born with the surface antigen type A, that means you have a type A blood. That's not going to cause your body to produce antibodies for it. So other people, they're born with a different type of antigen. They're going to be born with the antigen that's going to be type B here. So if there's type B. So the little blue triangles. So if you get a blood transfusion and let's say you are type A and then somebody accidentally gave you a blood type transfusion of type B. So what's going to happen is that you're going to put they're going to put B blood into your body, and somebody who's type A, they have antibodies to fight this type of antigen. So the triangular shape. So this is what's going to happen. It's going to cause agglutination or clumping and eventually break down, which is called hemolysis. So your body's going to start fighting type B blood because your body has antibodies to fight B. And then it's going to cause it to clump up and it's going to be fatal. So getting a blood transfusion of the wrong type is really bad. And the same thing happens if you do the opposite. If you go ahead and get a transfusion of type A when you're type B, then you're going to have antibodies for it that can go attach here, cause clumping, and then hemolysis. So I'm gonna let that sink in. So you have if you have the opposite antibody, depending on what blood type you have, it's gonna cause clumping. So let's go ahead and mix it up a little bit. So if someone's born with type AB, that means this antigen is part of their red blood cells, surface antigens. You have both of these antigens, you have both of these markers, antigen A and B. And since you have both of these markers, you're born with both of these markers, your body does not have any antibodies for them. Somebody who's born type E, you're missing the antibodies. You don't have any antibodies for the type A or B. So that means this person is going to be able to accept blood from anybody. If you give this person a transfusion of type B, no problem, because they already have type B antigen on their red blood cell. And if you give them a type A, that's also not an issue. They can accept that as well. So they don't have any antibodies to fight these. So you can also give them type O, because type O here don't have any antigens. So therefore, AB is considered to be universal acceptor. So this blood type is universal acceptor. That means if you have AB blood type, you can take any other kind of blood and it won't cause any problems. The last one we have here is type O. It has no markers on it, so that's going to be type O. This one has no surface antigens on there. Therefore, if you gave it, a blood A or B, that's a bad news. That means they have antibodies for type A and they have antibodies for type B. So they cannot accept any of the other types. So It doesn't recognize antigens, so these antigens will cause clotting. If you give it type B, that's also bad news because it doesn't have any B antigens. It'll clot, and obviously AB will be bad news too because it has both of these surface antigens. Or it has antibodies against both of the surface antigens. Therefore, type O has both of A and B antibodies. They can go ahead and fight A and B type. So on the other hand, um, if you give type O, because it doesn't have any surface antigen to, if you donated that to anybody, you can donate it to type A, type B, or type AB. It can be accepted. So we call this guy universal donor. This one is the reversal donor, so it can be donated to anybody because it doesn't have any surface antigens. This is the reason why whenever there's a call for blood donations, they really, really want people type O, because it doesn't matter if it's type O positive or O negative. You can just go ahead and donate, because in emergency, they're just gonna go straight for type O blood and they give it to a person. And there might be a reaction if it's a other type of blood and you didn't have time to do the blood typing. But if it's a planned surgery or something like that, then they have time to go ahead and plan it. They'll go ahead and type you, and they will give you the blood transfusion of the same type of blood. But if it's an emergency, then they don't have time, then they would try to give type O. So that's blood types. So next thing we have is Rh factor. So we said that the blood can be positive or negative. So it could be positive or negative, B positive, B negative, A positive, A negative, AB positive, AB negative, or O positive or O negative. So where's that negative and positive coming from? So that is positive is part of uh, positive and negative is coming from another antigen that's known as Rh antigen. So Rh antigen is named after this monkey, and this is a rhesus monkey. So that's where Rh stands for, and sometimes it's called D antigen. I'm not sure why they skipped C, but they just went straight to the D. And D antigen is the same thing. It's considered to be just another antigen on the surface of your blood. When you have this, for some reason, people who are blood type A and B, they don't have any antibodies for Rh. Um, so that means you're not born with it. So Rh factor is another antigen found on red blood cell surfaces. But I want to say that people are not born with the Rh antibodies. You're not born with it, so that means if you want to fight the Rh antigen, your body has to make the antibodies. So you're not born with this Rh antibodies. Your body has to go out and actually make it. That means, if somebody is Rh negative, they can accept, let's say, O positive blood. So Rh negative uh, can accept O positive blood. So let's think about a statement here. If you're going to be Rh negative, that means you do not recognize Rh antigen. Rh antigen is not recognized by your body. So because your body is not born with it, with the antibodies for it, you can go ahead and get blood transfusion of positive. Means you have, you can receive blood type that has the antigen. You will survive because you don't have Rh antibodies for it. On the other hand, now, once you've been exposed to Rh antigens, that'll cause your body to produce the antibodies for the Rh antigen. And what that's going to do is it's going to stimulate the body of Rh negative person to make Rh antibodies. So it might take a couple days or weeks, but eventually that person is going to, if someone is exposed to Rh antigen, they're going to make Rh, their body's going to produce the antibodies for it in the future. If they were, so If they were given the blood transfusion in the future with RH on it, then there's going to be problematic because their body's now full of these antibodies that'll fight it. So with that in mind, we're going to talk about how this works in the pregnancy. So again, people are not born with the RH antibodies, but if they are given a blood transfusion that's RH positive, they can accept the blood the first time, and because their bodies don't have the antibodies for it, but their body will start making the antibodies for it. So in the second time, it will be a problem. So with that said, with that in mind, let's see what's happening here. So we need to counsel women who are RH negative when they're pregnant because there's a problem or there's a possible disease that can happen. So if the woman has a child that's RH positive, so the important key components here are the fact that you have to understand is that it's only going to affect the females that are RH negative. So it's gonna affect the females that are RH negative. And if you're RH positive and you're pregnant, none of this affects you. So women go through prenatal care. That's one of the first things they look for is what your blood type is. So if you're going to be RH negative, next thing they're going to do is they're going to take a look at the baby and see what the baby blood type is, if it's a RH positive or RH negative. So then you might be wondering, how is it possible for a baby to be RH positive if the mother is RH negative? Now you'll have to go back and think about genetics. If you remember Mendelian genetics, you have two chromosomes. Well, you can inherit RH positive from the father. So the origin of the RH positive would be from the father that gives the gene the baby to be RH positive. So what's happening here, that in the beginning, the blood supply between the mother and the baby is separate. So there's no mixing of the blood between the mother and the baby. Everything is fine. But what happens when you go through childbirth is there's a mixing of blood. So there's lots of bleeding. Baby's blood is now blood is going to mix with mom's blood now, and then baby's blood mixes with mom's blood during labor. And remember, we said that if you expose RH negative person to RH antigen, then that's going to cause the body to produce antibodies for it. So when you have the first pregnancy, nothing bad will happen because mom doesn't have any antibodies for the RH antigen to fight the baby. So childbirth is relatively quickly, the baby comes out, everything's just fine. But in between, if there was mixing of blood happened, then the body starts making antibodies for the RH antigen. And now if that person was to have a second baby and the baby was RH positive and the mother had the RH antibodies, then mother's blood can attack the baby. Because she got exposed to RH antigen in the first place, if she were to be pregnant with the second child, the RH positive child, the RH antibodies can be fatal for the baby, and then they can start attacking the baby, start causing the blood to clump up. So that's how they respond. So that's anti-RH antibodies from mother cause hemolytic disease of newborn RH positive baby. So that's what it's called. The name of the disease is called hemolytic disease of newborn RH positive baby. So that's pretty common thing that could happen during pregnancy. And if you stay in the health field, you're probably going to counsel a woman that has to, go through the situation where the doctor didn't have enough time to explain it properly, and then you might have to do it. So the woman, didn't know what it means, then you have to explain how this disease works. So what happens in this scenario? So when you're RH negative and your baby's RH positive, you can take something called RhoGAM, this chemical. This is called RhoGAM, and it's an antibody itself. So this ROGAM is an antibody. So what it does is it'll go ahead and start binding to all these antibodies. It'll basically go and start binding to Rh antibodies, takes away all those antibodies and from the mother, and then Rh positive baby will not be in danger anymore. But it's only if you identify the disease early enough and you get the treatment. And the treatment is this chemical called ROGAM. So it can be prevented by giving mother ROGAM, is anti-Rh antibodies during pregnancy and at delivery. Okay, so the last thing I want to discuss here is what happens when you have a damage to your blood vessels or your tissues. So maybe this is basically if you have a cut or if you have damage to your blood vessels, the repair happens in 3 stages. The first one is called vascular spasm. And this is a smooth muscle contraction. So smooth muscle contracts and pushes away blood. That's vascular spasm. It causes, it does that to stop the bleeding. So the blood vessels, in the blood vessels, you have the smooth muscle we learned in last lecture, and it's going to kind of squeeze and kind of push the blood away. That's what happens when somebody turns white. If you see blood, um, you know, their face turns white, they might faint because their body went into vascular spasm. So if they were bleeding a lot, that means, and their face turned red and they kind of fainted. They went into vascular spasm because the blood shunted away from the surface of your face. And that's the first thing that happens. Next thing that happens is platelet. They come into action over here. So now your platelets, they come into action. And once they form the blood clot, then everything kind of coagulates. Coagulation is, again, kind of clumping, agglutination, a little bit similar. So it creates this clot. Coagulation is creating the clot to stop the bleeding. So once it's been repaired, you're going to have macrophages come in and get rid of the blood clot, and everything kind of goes back to normal. So those are the 3 stages. So if that went too fast for you, this is kind of what it looks like. So you have a damage to the blood vessel here, vessel injury. You're losing blood, and then vascular spasm happens. The smooth muscle kind of contracts and reduces the blood flow. So you can see this net-like structure going on. That's the smooth muscle. And you got these platelets that start to create the clot here. And it turns into a clot, it continues to repair, and eventually the clot goes away. And if your body spontaneously produces the blood clots, that's also bad. That's because it can break off, then go to your circulatory system, get caught, and can get in your brain or your spine or your lungs, and that'll be a stroke. So we don't want that to happen either. So we don't want the body spontaneously producing these clots. So this whole process is written for you step by step here, in case I went too fast for you. So you got this handout in Canvas. So what happens is that you have the damage to blood vessels. What I didn't mention was that there's this chemical, and that's called prothrombin activator. And that prothrombin activator converts prothrombin to thrombin. So from a plasma protein to an active enzyme. And then there is another chemical called fibrinogen. This is another soluble plasma protein that converts to fibrin. Once you have fibrin, that's what forms the clot. So let's analyze this. So you got these fibers. Fibers are what makes the plug. So if you ever took a scab off, maybe from maybe you scraped your knee and you had a scab fall off, and if you take the scab and look at it under the light, you're going to see these little lines. So let me see, let me go back. You're going to see these little lines, and these are fibers. So that's going to be fibrin that's generated by fibrinogen. the word gen stands for generating. So fibrinogen is the chemical that makes fibrin. So why do you want to have all these steps? Why don't you just have the fibrin straight up? Why do you have fibrinogen, thrombin, prothrombin activator? So it's because your body wants to make sure there's actually damage, and it wants to take off all these steps before it starts making clots, because like I said, if your body just spontaneously makes clots, that's not good either. So the prothrombin activator is a precursor, and it's going to activate the prothrombin, and that's going to activate the thrombin, and that's going to convert to fibrinogen, and that's going to go into the becoming a fibrin. So you have a bunch of these steps. Each one doesn't get activated unless the previous step is complete. So basically what I'm saying is that your body has created all these steps, all these extra steps, because they want to make sure you really are injured and you need to have blood clotting before you start making blood clots. So if you're clotting spontaneously, these clots can easily break off and then go and give you a stroke in your brain or your spine or your lungs. So that's really bad news. So you have basically a gene that makes prothrombinase, and another one that makes prothrombin, another one that makes fibrinogen. So you have all these genes for it. So in order to clot your blood, you need all these steps. So then the question is, what happens when you have a mutation if you don't have a gene for it? If you don't have a gene for let's say, make fibrin or fibrinogen, then you're not gonna make fibrin, and you're not gonna clot. So that means you have broken this chain now. If you break this chain, then you can't clot anymore. And people who cannot make clots, they have this disease that's called hemophilia, or a bleeding disorder. It's called hemophilia, or they're called hemophiliacs. So basically, when you have a mutation, you don't make one of these chemicals, you don't make one of those chemicals, then you get a cut, you just bleed out and bleed out, because your body it doesn't have all the things that it needs to make the clots. And this is also the origin of Dracula. So on the vampires, because back in the day, people didn't understand if you're a hemophiliac, it's just a genetic disorder. They thought that people were hemophiliacs or monsters because they had really pale skin, and that was obviously from bleeding a lot. And they got a cut, they'll bleed and bleed and bleed. They made up all these stories about people at nighttime, they'll go out, drink their blood. Philia means to be attracted to. So heme means blood, so they were attracted to blood. So there are all these crazy stories about Dracula and vampires. But in reality, it's just a genetic disorder. People are, missing one of those chemicals. They have a mutation, and then they are not able to clot. So if your hematocrit is too high, that's also bad. That means you know um, your blood is too thick. And some medications, they can interfere with hemostasis. That's called aspirin. So what the doctors recommend, if you have hematocrit, or high hematocrit that's too thick, then they will give you some baby aspirin to take every day, and that's going to thin out your blood. But if you don't have this, you have high hematocrit, you can take baby aspirin and they'll thin out your blood. But if you don't have that, instead you have hemophilia, then you're going to have difficulty clotting, and then if you take baby aspirin, in that case, that's gonna be a problem. So you might faint and things like that. So that's it for the blood chapter. Actually, that reminds me, Queen Victoria, she was a carrier for hemophilia. She had one of the genes for hemophilia, and she passed it on to lots of her children. So males, they have only one X chromosome. So if they have the chromosome that's affected, they are going to be affected, whereas females carry two X chromosomes. So if they only had one gene that was affected, they were carriers for it. And then look how many children she had. So I guess maybe she stayed pregnant um, because she was a carrier for hemophilia. So people who are carriers, they can still bleed a lot. They have heavy menstrual periods, and after surgery, they bleed a lot. So I guess she stayed pregnant most of her lifetime. That may have helped her avoid the heavy menstrual periods because she was a carrier for the hemophilia. So that's it for the blood chapter. Thank you guys for listening.